Hello, good evening, and welcome to all of you. This is Rajesh Kumar Singhal, and I am here to present a new topic on the stress protein. So, friend, this is one of the important topic, and in this lecture, we will discuss the one of the stress protein that is known as Lia protein, and in this lecture, we will discuss the structure, structure, functions, and types of the Lia protein. Okay, so this is important for the ARS men's exams. Okay, so let's start our discussion with the what is stress protein. Okay, so stress protein are the proteins. Okay, these are the functional proteins. These are the functional proteins which reduces the damage. Okay, so damage due to the abiotic stresses. Okay, also it protects from the abiotic stresses. So we can define the stress proteins that these are the functional proteins which reduces the first damages due to the abiotic stresses, and second they protect from the abiotic stresses. So this is stress proteins. Now come to the Lia proteins. Okay, so this is one of the stress protein which reduce the damages and protect against the abiotic stress. So what is the meaning of the Lia? That is known as late embryogenesis abundant protein. So what is the full form of Lia? That is late embryogenesis abundant. So this can be asked in the pre-exams or MCQ type questions, that what is the meaning of the Lia? That is late embryogenesis abundant protein. Okay, so this protein was discovered in the 1981 by the scientist Dewar and Crowd. Okay, CROUD. Okay, so this protein was discovered in the 1981 by the Dewar and Crowd in the cotton plant or cotton seed during the study of the embryogenesis. Okay, so this Lia protein was discovered in the 1981 by the scientist Dewar and Crowd in the cotton crop and during the embryogenesis study. So the name given the late embryogenesis abundant means these proteins are synthesized in the late embryogenesis stage or seed maturation stage. Okay, however, these proteins are not only found in the seed, but they also found in the leaves, root and stem tissue. Okay. Even these proteins are not found in the plant, they are found in the microorganisms. Okay. They are also found in the microorganisms and found in the leaves, root, stem. Even they found in the chloroplast or plastid and mitochondria. Okay. However, these proteins are predominantly found in the nucleus and cytosol. Okay. So these proteins predominantly found in the nucleus and cytosol. But these proteins also found in higher amount in the desiccating tissues. Okay. Or tissue where the water potential is less. Okay. So these proteins mainly found in nucleus and cytosol was also present in the plastid mitochondria. Also they found in the not only seed but they also found in the leaves, root and stems, and they also found in higher amount in the desiccating tissue where the water potential is very less. Okay. So the Lea proteins are very stable proteins. Okay. They can stay in even boiling water. Okay. So they are very stable proteins. Also these proteins are hydrophilic in nature. So what is the meaning of hydrophilics? Means they are water loving. Okay. And also these proteins rich in the glycine amino acids. Okay. So glycine is the rich amino acids and also polar amino acids are also found in the structure of this Lea protein. So what is the stress protein? We have discussed that what is the stress protein. These are the functional protein that reduces damage and production under the abiotic stresses. Also they are found in the plants and different microorganisms, and this Lea protein is known as late embryogenesis abundant because these proteins are mainly synthesized during the seed maturation stages. Also this present in the leaves, root, stem, plastid, mitochondria. However, predominantly found in the nucleus and cytosols. And Lea proteins are very stable proteins. They can stay in the boiling water, and also these are the hydrophilic in nature because also they are rich in glycine amino acids, and also they are water loving, and they are rich in the polar amino acids. Okay. So this is about the Lea protein. Now come to the structure of the Lea proteins. So Lea proteins have mainly three types of structures. Okay. So first structure is the random coil alpha helix. Okay. So this is the first structure, that is random coil, means this is the structure random coil. alpha helix, and second one is the empipathic alpha helix, and third is the mix where both random and empipathic alpha helix are present. So Lia proteins are present in the three structure. There are three types of structure. First is the random coil alpha helix, and second is the empipathic alpha helix, and third one is the mixed helix. So this random coil alpha helix help or prevent the crystalline or they are means maintain the membrane. Okay, membrane structure. And second is the empipathic alpha helix, which provide the stability to the membrane. Okay, so these are the important function of this alpha helix. So there are three types: random coil, empipathic alpha helix और mix. So random alpha helix का काम क्या है? ये आपका जो स्ट्रक्चर है उसको क्रिस्टलाइजेशन होने से डिहाइड्रेशन के टाइम्स, in the dehydration it prevent the crystallization of the macromolecules. And second, the empipathic alpha helix means both hydrophilic and hydrophobic parts is available, which increase the stability of the membranes. Okay, now come to the classification of the Lia protein. So there are mainly 6 group of the Lia proteins. So these are known as group 1, group 2, group 3, group 4, group 5 and group 6. So this group 1 includes the D19, and the important feature of this group 1 is that this have 20 amino acids. Okay, repetition of 20 amino acids. Okay, so this is the special characteristics of group 1 Lia proteins. Okay, so this is the Lia protein classification. Okay, so there are 6 type of Lia proteins. So group 1 is D19 and it has 20 amino acids repeats, and the important work in the endosperm development. Okay, and second is the osmotic production. This is also known as Lia 5. Okay, now come to the group seconds. So this group mainly known as dehydrates. Okay, so these are mostly known as dehydrates, DHY and also D11. Okay, so what is the special feature of these proteins? These proteins have 15 amino acids, conservative sequence at C terminal. ओके, तो सी टर्मिनल पे इस पे क्या होता है, 15 एमिनो एसिड का एक कंजर्वेटिव सीक्वेंस होता है, दैट इज आल्सो नोन एज के सेगमेंट. ओके, तो दिस इज द टिपिकल फीचर ऑफ डिहाइड्रेट्स दैट दे हैव के सेगमेंट्स. एंड व्हाट इज द इंपॉर्टेंट फीचर्स? दे वर्क एज ए molecular chaperones. So what is the molecular chaperones? So these work in the folding of the फोल्डिंग ऑफ द प्रोटीन्स। तो क्या होता है, जब स्ट्रेस के टाइम जो आपकी प्रोटीन डीनेचर हो जाती है उसको फोल्डिंग काम का काम करता है, ठीक है। सेकंड काम क्या करता है, दिस इज वर्क एज ए डिफेंडिंग प्रोटीन्स, ओके। सो डिफेंडिंग अगेंस्ट द डिहाइड्रेशन, ओके। नाउ कम टू द group 3, okay. So group 3 also known as D7, okay. This is also known as लिया 4, okay, लिया 4। तो एक चीज ये देख लेना, जो लिया 4 एंड ग्रुप 3 ये डिफरेंट है, जैसे G1 लिया 4 लिया 5, ओके, तो ये डिफरेंट है। तो G3 मीन्स लिया 4 एंड व्हाट इज द टिपिकल फीचर ऑफ दिस हैव 11 amino acids in 13 times repetition, okay. So 11 amino acids are repeated 13 times and form the empipathic alpha helix, okay. So what is the important function of this? The important function of this group 3 is the enriching ion or sequestration of ions, okay. So this is the important function of G3. Now come to the G4. So G4 is also known as Lia 1 or D113, okay. So this, what is the typical feature of this? This hair devoid of repeated motive sequence, means there is devoid of repeated sequence and conserved sequence at एंड टर्मिनल, जब किसमें किस पे था, सी टर्मिनल्स पे, ओके। तो दिस इज आल्सो फ्रॉम द एम्पीपैथिक अल्फा हेलिक्स, ओके। सो दिस G3 and G4 form the empipathic alpha helix and it increase the membrane stability, okay. So this is the function of this group 4. Now come to the group 5. So this is known as D34, also Lia 4. So the group 3 and group 5 include the Lia 4 and this also include D29 protein. So the important work is the dehydration tolerance, okay. And sixth group, that is also known as PB, Lia 18 or Lia 6, also include the SMP, that is the seed maturation proteins, ओके। तो पता चल रहा है नाम से। सो क्या करते हैं, दिस प्रोटीन प्रोवाइड द प्रोटेक्शन ड्यूरिंग द सीड मैच्योरेशन। सो फ्रेंड, दिस इज द important for classification of the Lia proteins, that there are 6 different groups, that G1, and they are different example, that G1 have D19, G2 have D11, G3 have D7, G4 have D113, G5 have D34 and D29, and G6 have Lia 18 and Lia 6. And this protein group 1 have 20 amino acid repeat sequence, G2 have K segment, that is 15 amino acid conserved sequence at C terminus, and G3 have 11 amino acid repetition in 13 times, and G4 devoid of repeated motifs, okay. So these are the important structures and this provide endosperm development and osmotic production. Group 2 work as a molecular chaperone and defending against the stresses. G3 work as a sequestration of ions. G4 provide the membrane stability. G5 provide the dehydration tolerance, and G6 are important as a seed maturation protein. So friends, this is about the classification of the Lia proteins. Now come to the important functions of the Lia proteins. Okay. So first function is that it work as a membrane stabilization. Okay. So it provide the stability to the membranes. Okay. Because this Lia protein have many hydroxyl radicals and form the hydrogen bond. Okay. So it provide the stability. Second, because due to amphipathic nature, it work as a hydration buffer. Okay. So this Lia protein work as a hydration buffer. What is the third important functions? Third important functions as a membrane protectant. Okay. So it provide the protection to the membrane. What is fourth functions? Fourth function, it work as a antioxidant or reverse scavenger. Okay. The fifth function is that it prevent the inactivation and aggregation of the proteins and macromolecules. Okay. Then what is the next functions? It act as a ion sequestration. How it work as a ion sequestration? As already discussed, that it have many charged amino acids. Okay. So they sequester the ions. Okay. 7. You know that when the water concentration during the seed maturation, okay, seed maturation, it reaches to the 10%. Okay. So water concentration, where it reaches to the very less than the cytoplasm or cytosol become in the glassy phase. Okay. So there will be probability to inactivation or dysfunctionalization of the organs. Okay. So at that time, the accumulation of this Lia proteins increase the density, okay, density of this phase and protect from the inactivation or dysfunctionalization of these organs. Okay. Also, these are the some important functions of the Lia proteins, and there are some important examples of Lia proteins. Like there is one example, that is core 15. This is Adopsis Lia proteins, and core work in the freezing tolerance. Then there will be a one Warli HbA 1 proteins, okay, protein that work under the osmotic or salt stress, okay. So these are the some important core proteins, okay. Now that this Lia protein can be synthesized by the three types, okay, by the ABA dependent, okay. So how they will form? By the exogenous and endogenous ABA concentration. So these Lia protein are dependent on the exogenous and endogenous ABA. Second, these are the induced ABA induced, okay. So when the stress, there will be a stress condition, increase in the ABA, and this ABA induce the synthesis of the these proteins, okay. And third one is the ABA independent, okay. So third one is the ABA independent. In this some this was studied in the ABA mutant. This is ABA mutants, okay. So in this, the accumulation of the synthesis of some gene, that's RD29A under the dehydration tolerance, okay, they increase the expression of this ABRE, that is abscisic acid response elements. These are the cis elements, and this increase the expression of Lia protein. So this is ABA independent. So friend, in these lectures we will discuss that what is Lia stress proteins, what is Lia proteins, and also we discussed the functions, structures, and basics of the basic information about the Lia proteins. So hope you understand this lecture. Thank you very much.